Imagine a world where the secrets of life itself lie not in ancient scrolls or divine texts, but in a microscopic structure found in every living organism. This structure is DNA, or deoxyribonucleic acid, the fundamental building block of life. But what exactly is DNA? DNA consists of two long strands forming a double helix, resembling a twisted ladder. Each rung of this ladder is made up of nucleotide pairs, specifically adenine with thymine and cytosine with guanine. These pairings encode genetic information essential for everything from eye color to susceptibility to disease. The history of DNA begins in the early 20th century, notably with the discovery of its structure by James Watson and Francis Crick in 1953. Their groundbreaking work laid the foundation for modern genetics, leading to the Human Genome Project, completed in 2003, which mapped the entire human genome comprising over 3 billion base pairs. With this massive data set came a plethora of information about the human body, but also a flood of questions regarding ethics and implications. As we delve deeper into the world of DNA, we realize its impact is not limited to human health. Consider how plants and animals utilize DNA. Genetic variations affect everything from crop yields to animal behavior. For example, the introduction of genetically modified organisms, GMOs, into agriculture has sparked significant debate. In 1994, the Flavre Sevier tomato became the first commercially grown GMO. It was engineered for longer shelf life and better taste. This case opened the floodgates for more GMO products, leading to higher crop resilience but also sparking fears about environmental and health effects. The power of DNA is immense, and as we explore its secrets, we encounter a crucial question. As we gain the ability to manipulate this blueprint, what moral responsibilities do we hold? What happens when we push the boundaries of genetic engineering too far? The possibility of engineering organisms, including humans, raises ethical concerns. Imagine a future where children are designed not by chance but by choice. The implications are vast and unsettling. This question of morality will echo through our exploration today, as we confront the potentials and pitfalls of genetic science. As we close this chapter, we're left pondering, what does it truly mean to be human? Stay with us as we navigate through the intricacies of genetic engineering in the next chapter. The advancements in genetic engineering technology have been nothing short of extraordinary. One of the most significant breakthroughs that has taken the scientific community by storm is CRISPR-Cas9. This revolutionary tool allows scientists to alter DNA with remarkable precision, and it has transformed molecular biology. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, works by utilizing a specialized enzyme called Cas9 that acts like molecular scissors. This system was originally part of the immune defense mechanism in bacteria, allowing them to fend off viral attacks by cutting up foreign DNA. Scientists adapted this natural process for gene editing, and it has since been harnessed to edit genes in a wide variety of organisms. In 2012, Jennifer Doudna and Emmanuel Charpentier published their groundbreaking work on CRISPR, which has since earned them numerous accolades, including the 2020 Nobel Prize in Chemistry. Their discovery has opened up countless avenues for research and potential therapeutic applications, from curing genetic disorders to combating diseases like HIV and sickle cell anemia. Consider the case of sickle cell anemia. This debilitating genetic disease affects millions worldwide and is characterized by misshaped red blood cells. Researchers are now using CRISPR to directly modify the genes responsible for this condition. Some patients have experienced remarkable recoveries, with their symptoms significantly alleviated after treatment. This represents an astonishing leap toward curing what was once considered an incurable disease. However, as we delve deeper into the realm of genetic engineering, we must confront the ethical concerns that accompany these advancements. For instance, if we can edit human DNA, should we? The prospect of designer babies becomes increasingly feasible. Parents might one day have the ability to choose traits for their children, from physical attributes to intelligence. Such capabilities challenge our concept of individuality and nature. Regulatory frameworks have struggled to keep pace with these rapid developments. Currently, some countries embrace genetic engineering, while others impose strict limits. The debate often pits innovation against moral concerns, with advocates emphasizing potential health benefits and critics warning of unforeseen consequences. As we explore further, we will discuss real-world implications and the ongoing debate surrounding genetic engineering. Will it herald a new era of medical miracles, or will it lead to ethical quagmires that humanity cannot escape from? In the next chapter, we will dive into these ethical dilemmas, highlighting stories that make us question the future of genetic manipulation. As we peer into the future of genetic engineering, we must grapple with the ethical dilemmas it presents. The capacity to edit human genes raises profound questions about the nature of humanity, consent, and the potential for inequality. Are we playing God by editing the very essence of our existence? One of the most significant ethical concerns revolves around the idea of designer babies. The very thought of selecting traits for future generations raises alarm bells amongst ethicists and scientists alike. If wealthier families can afford genetic enhancements, will we create a society divided by genetic privilege? This notion invokes comparisons to eugenics movements in the early 20th century, which led to horrific consequences in the name of improving humanity. The case of He Jiankui, a Chinese scientist who claimed to have created the world's first genetically edited babies, sent shockwaves through the scientific community. In 2018, he announced the birth of twin girls whose DNA had been altered to resist HIV infection. While he aimed to demonstrate the potential of gene editing, the world condemned his actions for lacking ethical oversight and transparency. This incident ignited a firestorm of debate about the responsibility of scientists in manipulating human life. Additionally, the potential for unintended consequences looms large in genetic engineering. Every intervention in the genome could have ripple effects, possibly leading to new health issues or exacerbating existing ones. For example, attempts to eliminate genetic disorders might also inadvertently introduce new mutations or destabilize other critical genes. The complex interplay of genes means that a seemingly small edit could have profound effects. Beyond individual cases, the implications extend to biodiversity as well. Editing the genes of certain species might result in ecological repercussions. Imagine altering a species to make it resistant to disease, but this change could inadvertently disrupt the ecosystem's balance. Such actions could lead us down a path from which there is no return. A pivotal question arises: who holds the moral authority to decide which genes to edit? The intricate web of morality and sociology must intertwine with the science of genetics. Engaging the public in discussions about genetic engineering becomes paramount. We need a collective voice to navigate these uncharted waters. As we sift through the ethical implications of genetic engineering, we are left with countless dilemmas to ponder. Stay with us as we explore the real-world applications of genetic therapies in the next chapter, examining their life-changing impacts and the stories that accompany them. As we continue our journey through the realm of genetics, we arrive at a pivotal application of genetic engineering: genetic therapies. These cutting-edge treatments are revolutionizing medicine by offering novel solutions for conditions once thought incurable. From rare genetic disorders to cancer, the potential is vast and transformative. One of the leading examples of genetic therapy is CAR T-cell therapy. This innovative approach involves reprogramming a patient's T-cells to seek and destroy cancer cells. By using a virus to modify these immune cells, doctors can enhance a patient's natural defenses against cancer. This therapy has shown remarkable success in treating specific types of blood cancers, such as leukemia and lymphoma. In a notable case, a young boy named Alex received CAR T-cell therapy after traditional treatments failed. His journey was fraught with challenges, but the therapy ultimately led to remission, providing a glimmer of hope to his family. Such stories exemplify the potential of genetic therapies but also highlight the emotional toll of battling life-threatening diseases. Beyond cancer, genetic therapies are making strides in treating rare genetic disorders. One noteworthy case is that of spinal muscular atrophy, SMA, a severe genetic condition affecting motor neurons. In response to SMA, a groundbreaking drug named Zolgensma was developed, designed to replace the missing or defective gene responsible for the disorder. With the potential to alter the course of the disease, this therapy has given countless families hope in what was previously a terminal diagnosis. However, these groundbreaking treatments come at a steep price. The cost of therapies like Zolgensma can exceed $2 million, raising questions about accessibility and equity. Can we truly call these advances revolutionary if only a select few can afford them? The healthcare system must grapple with the fairness of distributing life-saving treatments in an unequal world. As we witness these medical advancements, the emotional narratives entwined with genetic therapies remind us of the human side of science. From the joy of remission to the heartbreaking stories of loss, the stakes are undeniably high. Yet, these therapies also illuminate the ethical landscape, urging us to consider how we can ensure fair access to groundbreaking treatments. In the next chapter, we will explore the future of bioengineering, considering how genetic engineering might evolve and shape not just our health, but the entirety of life on Earth. As we round off our exploration of genetics, we gaze into the future of bioengineering, a frontier poised to redefine life on our planet as we know it. The possibilities are not just fascinating, they are potentially world-changing. Imagine a world where genetic engineering allows us to adapt to climate change, combat diseases, and even enhance our food supply sustainably. Synthetic biology, an emerging discipline, is making waves by enabling scientists to design and construct new biological parts. This innovative field seeks to create organisms with predefined functions. For instance, researchers are engineering bacteria to produce biofuels or generate pharmaceuticals. By harnessing the power of microorganisms, we can reduce reliance on traditional manufacturing methods, all while minimizing environmental impact. Such innovations could lead to greener alternatives to fossil fuels and create sustainable agricultural practices. Furthermore, consider the potential of genetically modified crops designed to withstand harsh climates or pest resistance. Crops like genetically engineered drought-resistant maize are being developed to thrive in conditions worsened by climate change. Farmers who integrate these crops into their systems could maintain productivity even in adverse conditions, potentially solving food scarcity issues around the globe. Yet, these advancements come with their own set of risks. The introduction of genetically modified organisms into ecosystems necessitates rigorous assessments. Unintended consequences may arise, and ethical questions linger regarding the manipulation of life forms. How do we balance innovation with responsibility? The future of bioengineering is not solely a scientific challenge, it is a moral one that requires careful navigation. As we peer into the not-so-distant future, we also encounter advancements in gene editing technologies. Researchers are exploring new tools beyond CRISPR, such as prime editing, which offers improved precision and flexibility in modifying genetic sequences. This evolution in technology promises to make gene editing safer and more effective, amplifying its potential applications. As we conclude this chapter, we are left to ponder profound questions about our place in the world. How will genetic engineering shape our identities and the universe around us? As we stand on the brink of unprecedented possibilities, we must engage in thoughtful dialogue about the future we want to create. The journey through DNA and genetic engineering is far from over, it is only just beginning. In our next chapter, we will summarize what we've learned and consider the responsibility that comes with such powerful knowledge. Imagine having the ability to edit the very fabric of life itself. With CRISPR technology, this once fantastical idea has become a reality. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, has taken the scientific community by storm. Developed in the early 2010s, it allows researchers to make precise alterations to DNA sequences, potentially eradicating genetic diseases and enhancing crop resilience. This chapter will delve into the mechanics of CRISPR, its applications, and the seismic shifts it is causing in genetics. Let's start with the basics. CRISPR works by employing a protein called Cas9, which acts like molecular scissors, cutting DNA at desired locations. Scientists design a guide RNA that directs Cas9 to the specific part of the genome they wish to alter. Once the DNA is cut, the cell's natural repair mechanisms kick in. Researchers can either disable a gene or insert new DNA to achieve desired effects. One of the most groundbreaking applications of CRISPR has been in agriculture. For instance, scientists have modified soybeans to make them resistant to drought, ensuring food security in changing climatic conditions. In one experiment, researchers at UC Berkeley introduced a gene that increased the drought tolerance of crops by up to 20%. Imagine the impact of such technology in regions facing food shortages. It offers the hope of sustainable farming amidst the growing threat of climate change. However, the revolutionary potential of CRISPR isn't confined to plants. In the realm of human health, CRISPR has shown promise in treating genetic disorders like sickle cell disease and muscular dystrophy. The first patient was treated in 2019 when doctors at the University of California, San Francisco utilized CRISPR to edit the genes of a patient suffering from sickle cell. Preliminary results have been promising, with patients showing significant improvement. While the prospects are exciting, they also raise profound ethical questions. Are we ready to manipulate human DNA? What if the edits lead to unforeseen consequences? A real concern arose in 2018 when a Chinese scientist claimed to have created the first genetically edited babies, sparking global outrage and debate. This chapter will explore not only the triumphs of CRISPR but also the inherent risks it poses to humanity's genetic legacy. What does it mean for the future of our species? As we dive deeper into the implications of CRISPR, it's essential to consider whether we have the wisdom to wield such power responsibly. The journey of gene editing is still unfolding, and the outcomes remain uncertain. What lies ahead could redefine the very essence of life itself. The concept of designer babies, children whose genetic traits are selected or modified, once lived in the realm of science fiction. Today, thanks to advances in genetic engineering, this prospect is inching closer to reality. This chapter will explore the ethical implications surrounding this controversial topic, from the potential benefits to the dangers of creating a genetic divide in society. Imagine a world where parents can choose traits for their children: intelligence, athleticism, even physical appearance. With CRISPR technology and other gene editing techniques, this vision could become a reality. The allure is undeniable, particularly for parents wanting to eliminate genetic disorders and enhance their children's prospects. A survey conducted in 2019 revealed that over 70% of parents would consider genetic interventions to prevent inherited diseases. However, the implications of such choices are profound. The question arises, who decides which traits are desirable? As genetic technology becomes more accessible, there is a risk that only affluent families could afford to enhance their children, creating a genetic caste system that could deepen social inequalities. This fear echoes historical eugenics movements that sought to improve the human race through selective breeding, often with horrific consequences. Experts fear that designs for better babies could lead to the loss of genetic diversity, which is crucial for the health of populations. Moreover, the long-term consequences of editing human genomes remain unknown. What happens if a genetic modification results in unforeseen problems generations down the line? An alarming study from the University of Pennsylvania highlighted potential off-target effects from CRISPR, suggesting unintended edits could occur in the genome, potentially leading to health issues. In this chapter, we will not only examine the biomedical and societal implications of designer babies but also the perspectives of bioethicists and scientists. How do we navigate the complexities of parental choice versus societal good? As we grapple with these questions, we must reflect on who we are as a society and what it means to be human. The quest for designer babies challenges us to consider the ethical boundaries of technology. Are we ready to cross those boundaries? The discussion is just beginning, and the stakes have never been higher. As the world grapples with biodiversity loss, could genetic engineering be the key to conservation? This chapter explores the fascinating applications of CRISPR and genetic engineering in wildlife conservation. From rescuing endangered species to combating invasive plants, genetic tools are revolutionizing the way we approach environmental challenges. One of the most notable examples includes efforts to save the woolly mammoth. Scientists at Harvard University have embarked on a project to bring back this iconic creature by editing the DNA of its closest relative, the Asian elephant. By introducing genes associated with cold resistance into the Asian elephant genome, they aim to create a hybrid that could survive in Arctic conditions. This project raises exciting possibilities but also concerns about ecological balance. What happens if these engineered creatures disrupt current ecosystems? In addition to de-extinction, CRISPR is being used to protect endangered species such as the Florida panther. A team of researchers at the University of California has been studying the genetic problems in this small population, looking for ways to introduce genetic diversity and combat inbreeding. By editing genes that increase the fitness of these animals, researchers hope to bolster their population and prevent extinction. Furthermore, genetic engineering is being utilized to combat invasive species that threaten native ecosystems. An example is the controversial project in New Zealand, where scientists aim to use CRISPR to eradicate invasive rats that threaten native birds. The goal is to edit the rats' genes to reduce their reproductive rates significantly. While promising, this approach raises questions about the potential ecological fallout. As we delve deeper into these innovative projects, we must weigh the potential benefits against ethical considerations. Will these advancements lead to unforeseen consequences in nature? How do we balance human intervention with natural evolution? The answers are complex and multifaceted, but one thing is clear: genetic engineering holds the potential not only to save species but also to redefine our role in the natural world. What does it mean for humanity to take on the mantle of Earth's genetic stewards? The path we choose could shape the future of biodiversity for generations to come. As we explore the frontier of genetic engineering, it's essential to understand its implications for our understanding of human evolution. In this chapter, we delve into how genetics have shaped our past and how modern technologies like CRISPR may influence our evolutionary trajectory. Genetic studies have unveiled an astonishing amount about our ancestors, offering insights into how traits evolved over millions of years. For example, ancient DNA extracted from Neanderthal remains has provided evidence that they interbred with early modern humans, leading to genetic contributions that still exist in people today. Researchers found that around 2% of the DNA of non-African humans is derived from Neanderthals, including genes associated with immunity and skin color. As we contemplate the role of CRISPR in future human evolution, we must ask, how will our understanding of genetics affect our species? Imagine a future where we can not only understand but also direct our evolution. This prospect could lead to enhanced cognitive abilities or increased resistance to diseases. However, it also raises concerns about the definition of what it means to be human and how society will perceive differences. Moreover, the relationship between our genetics and the environment cannot be understated. Epigenetics, the study of how environmental factors can affect gene expression, is revolutionizing our understanding of development and health. For instance, stress, nutrition, and lifestyle play crucial roles in which genes are expressed. This chapter will explore how our evolutionary history connects to genetic engineering today and the role environmental factors play in shaping our genetic legacy. The implications of genetic engineering stretch far beyond mere enhancement. As we take control of our genetic destiny, we must consider the ethical and philosophical ramifications. In this unprecedented era of genetic exploration, are we ready to embrace our role as architects of our own evolution? This chapter sets the stage for the momentous decisions we must face as we navigate the intersection of technology, identity, and evolution. As we conclude our journey through the world of DNA and genetic engineering, it's crucial to reflect on what lies ahead. The rapid evolution of genetic technologies presents us with both extraordinary opportunities and daunting challenges. What will the future of genetics look like, and how will it affect humanity? The ongoing advancements in genomics, particularly through machine learning and artificial intelligence, are promising groundbreaking insights into complex genetic interactions. These technologies enable scientists to analyze vast amounts of genomic data, uncovering patterns that were previously hidden. For instance, AI algorithms can predict patient responses to certain genetic therapies, personalizing treatment plans in ways that were unimaginable just a decade ago. However, with great power comes great responsibility. As we advance, the need for ethical guidelines becomes ever more pressing. Questions around consent, accessibility, and potential misuse of genetic data must be addressed to ensure that the benefits of these technologies are equitably distributed and do not deepen existing inequalities. A collaborative global approach will be essential to navigate the ethical landscape of genetic engineering. As we stand on the precipice of a genetic revolution, the future of CRISPR and gene editing technologies promises to change our lives in unimaginable ways. We need to contemplate not only the scientific possibilities but also the societal implications of our choices. Will we harness genetic technologies to enhance our health and well-being, or will they lead us into ethically murky waters? In conclusion, the journey through DNA and genetic engineering reveals a universe of possibilities. The decisions we make today will shape the generations to come, and it is our collective responsibility to guide the ethical use of these powerful tools. The future is unfolding before us, and how we choose to interact with the world of genetics can either elevate humanity or steer it into perilous territory. As we leave behind the stories of DNA, we must encourage an ongoing conversation about our genetic legacy, ensuring that we remain vigilant stewards of the future. What will we choose to create? The possibilities are vast, but the responsibility is ours. The dawn of genetic engineering has ushered in an unprecedented arms race among nations, researchers, and corporations. The question that looms is, who will dominate this field, and what are the implications for humanity? Countries like the United States, China, and the European Union are pouring billions into genetic research, each vying for breakthroughs that could lead to advancements in agriculture, medicine, and biotechnology. This isn't just about science, it's about power, economics, and the very essence of life itself. In recent years, China's investments into CRISPR technology have skyrocketed, often outpacing those of the United States. For example, in 2018, Chinese scientists made headlines when they announced the birth of the world's first genetically edited babies, Lulu and Nana, whose DNA was altered to resist HIV. The ethical outcry was immediate and fierce, fueling international debates. But what many missed was the broader context, an ongoing competition to harness the power of genetics for economic and political leverage. As nations race to claim supremacy, the impact is being felt in academia and industry. Partnerships and collaborations are forming across borders, aiming to share knowledge and resources. For instance, the Global Gene Editing Regulation Project, launched in 2018, aims to provide guidelines for safely using gene editing technologies worldwide. But even within collaborative frameworks, trust issues persist, often leading to tensions. Moreover, the commercialization of genetic advancements is a double-edged sword. While companies like Adidas Medicine and CRISPR Therapeutics are developing therapies for diseases, the question arises, will these innovations be accessible or only available to the wealthy? As gene editing becomes a commodity, the gap between those who can afford advanced treatments and those who cannot may widen, leading to social unrest and ethical dilemmas. This arms race thus brings not just competition but also accountability and moral responsibilities. As we look ahead, the implications of genetic manipulation are staggering. Will future conflicts be fought over genetic resources and patents? How can we ensure that advancements benefit humanity as a whole rather than a select few? This chapter serves as a critical reminder. As we unlock the secrets of the genome, we must tread carefully and thoughtfully, considering not just what we can do but what we should do. In closing, the race for genetic supremacy reveals much about human nature. It reflects our drive for progress, our ambition to dominate, and our ongoing struggle with ethics. With every gene we edit, we must ask ourselves who we are becoming. Will we shape our destiny responsibly, or will we lose our way in the allure of scientific grandeur? The answers remain to be seen as we continue this fascinating journey into the unknown world of genetics. As the global population surges past 8 billion, ensuring food security has become one of the most pressing challenges of our time. Genetic engineering has emerged as a potential game-changer in agriculture, promising solutions that could revolutionize how we grow our food. This chapter explores how innovations such as genetically modified organisms, or GMOs, are reshaping the agricultural landscape. In the 1990s, the introduction of Bt cotton marked a pivotal moment in agricultural biotechnology. This crop was engineered to produce a toxin from the bacterium Bacillus thuringiensis, which effectively kills certain pests. Farmers reported substantial increases in yield and reductions in pesticide costs, leading to higher profits and lower environmental impact. However, the success of Bt cotton also sparked numerous debates about the safety and ethics of GMOs. Critics argue that GMOs can lead to unforeseen ecological consequences, such as the development of resistant pests, loss of biodiversity, and potential health risks. In response, many scientists emphasize the rigorous testing protocols that GMOs undergo before reaching consumers, asserting that they are as safe as their non-GMO counterparts. Yet, public perception remains divided, often influenced by misinformation and fear. The ongoing struggle is to find common ground between scientific advancement and consumer concerns. Beyond crop yields, genetic engineering has opened doors for creating drought-resistant and salt-tolerant crops, essential for regions experiencing climate change. Research is underway to modify staple crops like rice and wheat, aiming to ensure food security even as the climate becomes increasingly unpredictable. The Golden Rice Project, engineered to contain vitamin A, is one such initiative aimed at combating malnutrition in developing countries. Yet, its rollout has faced criticism and bureaucratic hurdles, highlighting the complex interplay of science and policy. The introduction of CRISPR technology takes agricultural genetic engineering to new heights, allowing for precise modifications without the introduction of foreign DNA. Scientists have successfully edited the genome of crops to enhance their nutritional value and resilience, all while bypassing the lengthy regulatory processes tied to traditional GMOs. This method has the potential to revolutionize agriculture, but it also raises critical questions about regulatory frameworks and the ethical implications of such precise interventions. As we explore the future of agriculture, one thing is clear: genetic engineering will play a pivotal role in feeding the world's growing population. But we must navigate the ethical implications carefully, ensuring that innovations benefit all and do not exacerbate existing disparities. The challenge lies in balancing scientific progress with ecological sustainability and ethical responsibility. In conclusion, genetic engineering offers a beacon of hope amidst looming food crises. As we harness this powerful technology, we must remain vigilant stewards of the planet, ensuring that our quest for progress does not sacrifice the health of our ecosystems or our society. The answers we find will shape the future of agriculture and ultimately the fate of humanity itself. What if we could unlock the secret to aging? The quest for eternal youth has fascinated humanity for centuries, and recent advancements in genetics are bringing us closer than ever to understanding this complex biological process. In this chapter, we navigate the burgeoning field of aging research, where scientists explore the genetic underpinnings of longevity and the potential for life-extending therapies. One of the most groundbreaking discoveries in this field involves the enzyme telomerase, which plays a crucial role in maintaining the telomeres at the ends of chromosomes. Telomeres shorten with each cell division, leading to cellular aging and eventual senescence. Researchers found that activating telomerase could extend the lifespan of certain cell types, igniting hopes for anti-aging therapies. A groundbreaking study from 2020 demonstrated that mice genetically modified to express telomerase experienced not only extended lifespans but also improvements in health during aging. Imagine a future where these findings could translate into therapies for humans. Furthermore, studies on the genetics of centenarians reveal fascinating insights into resilience against age-related diseases. Certain genes seem to confer protective effects, allowing individuals to live longer and healthier lives. For example, the FOXO3 gene, found in many centenarians, is associated with longevity and reduced risk of age-related diseases like cardiovascular issues and Alzheimer's. Understanding these genetic factors opens the door for targeted therapies that could mimic these protective effects in the general population. But unlocking the secrets of aging isn't just about extending life, it's about enhancing the quality of life as we grow older. As researchers delve deeper into the genetic basis of aging, the focus is shifting toward developing interventions that maintain health and vitality. For instance, a promising area of research involves senolytics, a class of drugs designed to eliminate senescent cells, which contribute to age-related decline and inflammation. Early studies have shown that removing these cells can improve health span, the period of life spent in good health. Yet, as we stand at the edge of these discoveries, ethical questions emerge. Who gets access to anti-aging therapies? Could such treatments exacerbate existing inequalities in healthcare? Furthermore, what are the implications of significantly extending human lifespans on our social, economic, and ecological systems? The prospect of longer lives could fundamentally alter our perceptions of aging and the value we place on various life stages. In conclusion, the genetics of aging represents a new frontier in scientific research. As we dare to dream about living longer, healthier lives, we must tread carefully. Each breakthrough offers not just hope but a responsibility to reflect upon the ramifications of our actions. As we continue this journey into the intricate web of genetics, we stand on the precipice of profound transformation, one that could redefine the human experience itself. When we think about genetics, we often focus solely on DNA sequences, the letters of life that dictate everything from our eye color to our susceptibility to diseases. But what if I told you that there's a layer of genetic expression that goes beyond mere DNA? Enter the fascinating world of epigenetics. This science examines how environmental factors can alter gene expression without changing the underlying DNA sequence itself. Imagine a switch that can turn genes on or off, influenced by your diet, lifestyle, and even stress levels. In 2017, researchers discovered that a father's diet before conception could change the way genes are expressed in his offspring. This revelation has profound implications for understanding not only human development but also evolution and inheritance. Epigenetics suggests that our life experiences can literally change our DNA, influencing generations to come. For example, studies on the agouti mouse showed that when fed a specific diet rich in methyl donors, the expression of the agouti gene changed, leading to a spectrum of fur colors. These findings raise the question, could lifestyle changes help us combat genetic predispositions to certain diseases? Furthermore, epigenetic changes can also play a role in diseases such as cancer. Many tumors exhibit distinct epigenetic patterns compared to normal tissue. This understanding has propelled researchers to explore targeted epigenetic therapies. One of the most groundbreaking avenues is the development of drugs aimed at reversing harmful epigenetic modifications. For instance, in clinical trials, a drug named azacitidine showed promise in treating acute myeloid leukemia by restoring normal gene function. As we delve deeper into epigenetics, we uncover the potential for not only treating diseases but also enhancing health and longevity. The idea that our environment, thoughts, and behaviors can reshape our genetic destiny is nothing short of revolutionary. Will we soon be living in a world where we can alter our epigenome to prevent diseases before they even have a chance to emerge? In conclusion, understanding epigenetics broadens our perspective on genetics and health. It's not just about the DNA we inherit, but also about how we live our lives. The potential to manipulate these epigenetic mechanisms offers a new frontier in science, one that encourages us to consider the profound impact of our daily choices on future generations. But as we unlock these new potentials, we also face new ethical questions, raising the specter of who gets to access these advancements and how society will manage this knowledge. A new frontier in genetics lies within a groundbreaking technology called gene drives. Imagine being able to spread a genetic modification through an entire population, ensuring that a specific trait becomes dominant. Gene drives leverage the principles of inheritance in radical new ways, effectively hitchhiking desirable genes through generations. The implications are staggering. In 2015, scientists successfully introduced gene drives in laboratory populations of fruit flies, altering their genes to not only express a particular trait but to ensure that the modified gene is passed on to almost all of their offspring. This technique could revolutionize how we tackle pressing environmental and public health challenges. For instance, researchers are investigating the potential of gene drives to eliminate malaria. By modifying mosquitoes to carry a gene drive that leads to infertility or altered lifespans, we could drastically decrease their populations, thereby reducing the transmission of this deadly disease. However, the power of gene drives also comes with significant ethical and ecological considerations. What happens when a modified organism begins to propagate in the wild? The consequences of disrupting natural ecosystems could be catastrophic. The potential for unintended effects raises questions. Are we prepared to manage the consequences of these modified species? One of the most notable projects investigating gene drives is the Target Malaria Initiative. It aims to reduce malaria transmission through genetically modifying mosquitoes. Initial trials have shown promise, but they also face resistance from community groups concerned about the long-term impacts. As we push the boundaries of genetic engineering, the landscape of not just agriculture, but wild ecosystems, could fundamentally shift. The urgent need for effective solutions to combat vector-borne diseases has paved the way for gene drives to be viewed as a viable solution, yet we must tread carefully. The question remains, will we find a balance between innovation and responsibility? In conclusion, gene drives represent the dual-edged sword of modern genetic engineering. While they offer pioneering solutions to some of humanity's most pressing challenges, they also compel us to confront critical ethical dilemmas as we wield this powerful tool. As we voyage further into the genetic realm, the conversations we hold today will shape the world of tomorrow. Will we act with foresight and caution, or rush headlong into uncharted waters? What if we could not only edit life but also create it from the ground up? Welcome to the realm of synthetic biology, a revolutionary field that combines engineering principles with biological research to design and construct new biological parts, devices, and systems. Since the arrival of synthetic biology in the early 2000s, we have witnessed astonishing advancements. For example, in 2010, scientists from the J. Craig Venter Institute created the first synthetic cell, named Mycoplasma mycoides JCVI Syn 1.0. This landmark accomplishment demonstrated that we could build a living organism from a synthetic genome, composed entirely of chemical building blocks. The implications of this achievement are vast. Synthetic biology can lead to innovations in drug production, biofuels, and even pollution cleanup. One exciting application is engineering bacteria to produce pharmaceuticals. Imagine a world where microbes churn out life-saving drugs without the extensive resources currently required for pharmaceutical manufacturing. Furthermore, synthetic organisms can be designed to consume waste, breaking it down and rendering it harmless. Yet, as we explore this brave new world of synthetic life, ethical dilemmas arise. Who owns the rights to these synthetic organisms? Should we create entirely new life forms, or are there boundaries that should never be crossed? In 2021, the scientific community grappled with a significant event, the creation of a self-replicating synthetic organism that could evolve. This breakthrough raised alarms regarding biosecurity and the potential for unintended consequences in the environment. The fear of creating organisms that could outcompete natural species or disrupt ecosystems looms large. In the face of these challenges, the potential benefits of synthetic biology are immense. From combating climate change to curing diseases, the possibilities seem boundless. But with great power comes great responsibility. Balancing innovation with ethical oversight will be crucial as we venture further into this uncharted territory. In conclusion, synthetic biology stands at the frontier of life sciences, where engineering meets biology in unprecedented ways. As we learn to fabricate life, we must remain vigilant about the implications of our creations. The choices we make today will shape the relationships between humans and the very fabric of life on Earth. Will we be responsible stewards of this knowledge, or will we risk losing sight of the delicate balance of nature? DNA has always been heralded as the blueprint of life. But what if I told you that the story of genetics is much deeper and more complex than the mere sequence of nucleotides? Enter epigenetics, a fascinating field that explores how external factors influence gene expression without altering the underlying DNA sequence. It's a realm where environmental variables, lifestyle choices, and even emotional experiences leave their marks upon our genes. Imagine a child born into a family steeped in poverty. The stressors of that environment, poor nutrition, pollution, and social instability, may activate or repress certain genes, potentially affecting that child's health for generations. Compelling evidence from studies shows that environmental stresses can cause epigenetic changes, altering gene expression that dictate conditions like obesity, diabetes, and mental health disorders. For instance, researchers from the National Institute of Environmental Health Sciences have demonstrated how exposure to toxic substances can affect the genes of not just individuals, but their offspring too. This revolutionary perspective compels us to reconsider traditional views of inheritance and suggests a potential for biological and psychological inheritance that transcends generations. This raises incredible possibilities and enormous challenges. If our genes can be manipulated not just through editing, but through changes in our environment and lifestyle, what does that mean for the future of mankind? Should we regard our genetic makeup as fixed, or can we adapt our biology through conscious choices? Ancient wisdom has long suggested that we are the sum of our choices, but now, science is echoing this sentiment, particularly through the lens of epigenetics. In animals, there are striking examples of how epigenetics plays a crucial role in survival and adaptation. The famous case of the agouti mouse illustrates this point vividly. The agouti gene, when expressed, leads to a yellow coat, obesity, and a predisposition for disease. But what if these mice are fed a diet rich in methyl donors during gestation? The result is profound. Many mothers produce brown-coated offspring that are healthy and lean. This change is not a result of altering the DNA sequence, but rather the influence of nutrients on gene expression. It poses a compelling question: how might food choices and nutrition shape our genetic expression and well-being? As we delve deeper into this field, the implications extend beyond individual health into public policy and education systems. If our genes are susceptible to environmental influence, we may need to rethink approaches to healthcare, nutrition, and education. Should schools incorporate strategies that reduce stress among young students to support healthier gene expression? As we begin to grasp the immense power of epigenetics, a new narrative of human health emerges, one that champions prevention and holistic well-being. However, as with all powerful tools, this knowledge also brings with it ethical dilemmas. Should we manipulate our environments to alter gene expression deliberately? What are the potential long-term consequences of such actions? How do we ensure equitable access to these life-altering advancements? Gene drives are cutting-edge genetic technologies designed to propagate a particular suite of genes throughout a population. They offer solutions that could once only be imagined. However, with their power comes significant ethical considerations. Imagine being able to alter the genetics of an entire population, from eradicating invasive species to eliminating diseases like malaria. Gene drives function by ensuring that a specific genetic alteration is inherited more frequently than by the standard 50% Mendelian inheritance. This means a modified gene can spread rapidly through a population, bypassing the typical limits of inheritance. For example, researchers modified the genetic makeup of mosquitoes so they could not transmit malaria. Just a few generations later, nearly the entire population was resistant to the disease. This powerful technique demonstrates the potential to reshape ecosystems and conquer public health challenges. One of the most iconic examples of gene drive technology comes from the University of California, San Diego, where scientists engineered a strain of the common house mouse to carry a gene that would spread sterility among their populations. This could potentially help combat invasive species that threaten biodiversity. In another ambitious project in Brazil, scientists hope to spread a gene that will cause local populations of Aedes aegypti mosquitoes to self-destruct, drastically reducing the incidence of dengue fever and Zika virus transmission. Yet, with great power comes great responsibility, and the deployment of gene drives isn't without its risks. The potential for unintended consequences looms large. What if the modified gene escapes into non-target species? What could happen to local ecosystems? These questions bring us to the forefront of ethical and regulatory discussions around gene editing technologies. Regulatory regimes around the world are struggling to keep pace with these rapidly evolving technologies. Many argue that due to the potential irreversible consequences of gene drives, their use should be closely monitored or even prohibited. In 2016, a group of scientists held a public forum to discuss the ethical implications surrounding gene editing and gene drives. They highlighted the need for comprehensive public engagement and dialogue, recognizing that the implications of these technologies extend well beyond the laboratory. Social scientists and ethicists emphasize the importance of including local communities in the decision-making process, particularly when it comes to interventions that will impact the environment. Moreover, there are concerns about who controls these technologies. Large biotech companies are investing heavily in gene drive research, leading to fears of monopolization and unequal access. What happens to communities in poverty if they cannot afford these cutting-edge technologies that can save lives? As we tread into this uncertain territory, the question becomes, how do we harness the potential of gene drives while ensuring equitable access and safety? The stakes are high, and as researchers across the globe explore the potentials of gene drive technology, we find ourselves at a crossroads. The future may hold answers to long-standing human challenges, but we must approach this new frontier cautiously. This powerful tool teases a world where we can solve pressing issues, but at what cost? It is clear that while gene drives can be a beacon of hope, they carry the weight of ethical responsibility and demand a thorough examination of their implications, not just for humanity today, but for generations to come. How we answer these questions may very well define the trajectory of our shared future. Imagine a world where scientists can create life from scratch. This may sound like science fiction, but in the realm of synthetic biology, it is becoming a reality. Synthetic biology blends biology and engineering to design, construct, and manipulate new biological parts and systems. It presents both thrilling possibilities and unnerving questions. Are we playing God, or are we merely advancing human capability? The journey into synthetic biology began with the manipulation of DNA, but it's grown into a complex field that now encompasses the creation of entirely new organisms. One of the groundbreaking achievements in this field came in 2010 when scientists at the J. Craig Venter Institute created the first synthetic bacterium, Mycoplasma mycoides JCVI Syn 1.0. By assembling a synthetic genome and transplanting it into a bacterial cell, they essentially created life, proving that life's fundamental blueprint can be synthesized. Synthetic biology has vast applications, from medicine to agriculture and environmental sustainability. In medicine, researchers are designing bacteria with programmed abilities to detect and destroy cancer cells. Imagine being able to swallow a pill filled with engineered microbes that hone in on tumors and obliterate them while leaving healthy cells intact. This is not a distant dream, it's happening now. Researchers at MIT have pioneered using synthetic biology to program cells that can produce insulin, providing new pathways for the treatment of diabetes. In agriculture, synthetic organisms can help address challenges posed by climate change, such as drought and disease resistance. For instance, engineered crops can be designed to thrive in harsher environments, potentially reducing the need for pesticides and increasing food security worldwide. Companies like Indigo Agriculture are exploring ways to create crops that can sequester carbon from the atmosphere, contributing to climate change mitigation efforts. However, with such power comes responsibility. The manipulation of living organisms raises significant ethical concerns. Are we prepared for the consequences of creating synthetic organisms that could interact unpredictably with natural ecosystems? The potential environmental impact is enormous, and discussions about regulations governing synthetic biology are ongoing. In 2018, a controversial CRISPR experiment in China edited the genes of embryos to create genetically immune children, which sparked a global outcry about the ethical implications of gene editing and synthetic biology. The overarching question is, should there be limits to what we can create? How do we ensure safety and ethical governance in a field that is advancing so rapidly? The potential benefits of synthetic biology, from healthcare advancements to sustainable solutions for agriculture, are staggering, but they also pose existential risks. As we march into this uncertain future, we must ask ourselves whether we are prepared to face the implications of creating new life forms. As we continue to unravel the mysteries of genetics and synthetic biology, each discovery has the potential to redefine our understanding of life itself. In this ongoing journey, science pushes the boundaries of creativity and innovation, but we must tread carefully, balancing ambition with responsibility. The choices we make today can shape the world of tomorrow, and how do we ensure we do it wisely? What will our legacy be in a world where life can be crafted from scratch? Imagine if your environment, what you eat, how you live, and the stress you endure, could alter your DNA without changing its sequence. This incredible phenomenon is called epigenetics, the study of how external factors can influence gene expression. Scientists are discovering that epigenetic changes can be passed down through generations, posing profound questions about inheritance, identity, and evolution. Let's explore this fascinating field and see how it reshapes our understanding of biology. Epigenetic modifications occur through chemical changes that occur on DNA or the proteins associated with it, primarily involving molecules like methyl and acetyl groups. For example, DNA methylation can silence genes, while modifications to histone proteins can either activate or repress gene expression. These processes influence everything from how we grow to our susceptibility to diseases. A groundbreaking case study involves identical twins, Anna and Maria. Despite sharing the same DNA, their epigenetic profiles diverged significantly due to different lifestyles. Anna lived a stress-free rural life, while Maria faced chronic stress in an urban environment. Researchers found that Anna's genes associated with immunity were more robustly expressed compared to Maria's, revealing a compelling link between their surroundings and genetic expression. Such differences demonstrate that genetics is not solely deterministic. Rather, our lifestyle choices can have lasting impacts on our health and well-being. Moreover, epigenetics plays a crucial role in cancer development. Tumors can arise from abnormal epigenetic changes, leading to the silencing of tumor suppressor genes or the activation of oncogenes. Researchers are investigating targeted therapies that can reverse these modifications, presenting new avenues for cancer treatment. One remarkable example is the drug azacitidine, which reactivates silenced genes in some types of leukemia, providing hope where traditional treatments fail. Beyond individual health, epigenetics extends into agriculture. Crop resilience can be improved through epigenetic techniques, helping plants adapt to climate change. A study on rice showed how epigenetic modifications could enhance drought resistance, offering a solution to food security issues. As we delve deeper into epigenetics, it becomes evident that our identities and even our futures are intricately woven with these invisible threads. The implications are staggering. Could the choices we make today influence not just our health, but also the genetic destinies of our descendants? This questions the very fabric of evolution, where the interplay of genes and environment continually reshapes the potential of life. Epigenetics thus emerges as a transformative lens through which to view genetics, reminding us that biology is not a fixed narrative but a dynamic story. As we harness the potential of this knowledge, we may unlock revolutionary insights into health, evolution, and the essence of what it means to be alive. The journey of discovery continues, leading us toward a future where understanding our genetic heritage will empower us to make healthier choices for ourselves and the next generation. Imagine a world where scientists can ensure that a specific trait spreads rapidly through a population, potentially eradicating diseases or even certain species. This is the promise of gene drives, an innovative genetic engineering technique that leverages the power of CRISPR technology to bias inheritance patterns. As we stand on the brink of this new era, let's explore the science, potential benefits, and ethical concerns surrounding gene drives. Gene drives work by altering the rules of genetic inheritance. Typically, offspring inherit a parent's genes with a 50% probability. However, gene drives can increase the probability of inheritance to nearly 100%, effectively ensuring that a modified gene spreads throughout a population. This technology aims to address critical challenges like vector-borne diseases, where organisms like mosquitoes carry dangerous pathogens. One groundbreaking project involves the modification of the Anopheles gambiae mosquito, a primary vector for malaria transmission. By engineering a gene drive to render these mosquitoes sterile or resistant to the malaria parasite, researchers hope to reduce malaria infections dramatically. A field trial is currently underway in Africa, aiming to test the safety and efficacy of this approach. The stakes are high, millions of lives could be saved, yet the ecological consequences of releasing modified organisms into the wild remain uncertain. What happens if a gene drive inadvertently alters entire ecosystems? The case of the gene drive raises profound moral questions. Who decides if a species is allowed to be altered or eradicated? The technology holds immense power and requires rigorous ethical considerations. In response, conservationists and biologists are advocating for transparent discussions and strict regulations to ensure responsible use. Notably, gene drives are not only limited to disease control. They could also play a pivotal role in combating invasive species threatening native biodiversity. A study proposed using gene drives to reduce the population of invasive rodent species on islands, where they pose a significant risk to native bird species. By engineering a gene drive to ensure that the invasive species cannot reproduce, scientists could restore ecological balance. However, the unintended consequences of such actions must not be underestimated. Could we create another ecological catastrophe in our quest to correct a prior mistake? As we navigate the possibilities of gene drives, we must also weigh the potential societal impacts. There exists a fear that such powerful tools could be misused for malicious purposes, like creating bioweapons or enhancing certain traits in humans. This highlights the urgent need for global governance and ethical guidelines that keep pace with scientific advancements. In conclusion, gene drives represent a transformative tool in the arsenal of genetic engineering. Their potential to solve pressing global